Assalamualaikum. Today, my group would like to present about DNA replication. So here are my group members. So let's start. First of all, DNA basically controls the cell so that when you make a new cell, like in mitosis, more DNA is produced into the new cell. That's basically the surface of DNA replication, which also means making more DNA. Okay, so where and when is the questions now. So where? So replication occurs in the nucleus if the cells have nucleus, because we know that not every cells have nucleus, right? So we are now focusing on eukaryote cells that have nucleus, because prokaryotic cells actually does replication also, but in a different way. We are now focusing on eukaryote cells only as it have nucleus. Now the question when. When does this occur? It actually happens in the stage called interphase, where the cells grow and also carries out cell processes and of course replicating DNA. So do you guys want to know what it doesn't do? It doesn't divide. No cells are dividing, guys, in this stage because it can't replicate and divide at the same time. It is as if it's doing too much multitasking. So during cell division, more known as mitosis, no DNA replicating. In fact, cell replication is before the division process. This is because once we make a new cell, there's got to be a new DNA. Now I will pass to my friend Mastura to tell more about DNA replication.
Now I'm going to introduce you the key players of DNA replications. The majority of these key players are enzymes. When you see something that ends with ASE, it is very possible that it is enzyme. Enzymes has the ability to speed up reactions and build up and break down items that they add on. Let me introduce you the first key players. First, helicase. It is the unzipping enzymes. When you recall that DNA has two strands, you can think that helicase unzipping the two strands of DNA without having a hard time. DNA polymerase is a builder. These enzymes replicate DNA molecules and build a new strands of DNA. Primase is the initializer. DNA polymerase cannot figure out where to start it without primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. The interesting fact about primase is made out of RNA. Last but not least, ligase. Ligase is the glue. It helps to join DNA fragments together. Thank you.
Hello, my name is Muhammad Shafiq bin Hafiz. DNA replication starts at a certain part called origin. Usually this part is identified by a certain DNA sequences. There can be multiple origin within the DNA. At the origin, helicase comes in and unwinds the DNA. Single-stranded binding proteins aka SSB proteins binds to the DNA strands and they keep them separated. Primase comes in and makes RNA primers on both strands. And remember, that's really important because otherwise, when DNA polymerase comes in, it wouldn't know where to start. Then only the DNA polymerase can get to work, which is to add the DNA bases. Now we have two strands that is not identical. They complement each other. They also are anti-parallel. This means they won't go to the same direction. With DNA, we don't say a strand goes north or south. The strands for DNA strands are the directions for the DNA strands are a little different. We say that DNA either goes from 5 to 3, 5 prime to 3 prime or 3 prime to 5 prime. This seems very confusing, right? Because the sugar in the DNA is part of the backbone for a DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. The 5 prime carbon is actually outside the structure. When we do this on the other side, keep in mind that the strand is flipped because it's anti-parallel.
In DNA replication, at the elongation process, the new DNA strand is synthesized. The DNA polymerase III enzyme catalyzes the elongation of new DNA strand at the location after the primer by continuously adding nucleotides to the new complementary strand. There is two types of new complementary strand, which is leading strands and lagging strands. The leading strands are growing towards replication forks, which DNA primers can only go from 5' to 3' direction. This is because it can only add the nucleotides to the 3' end. Meanwhile, for the lagging strands, the DNA polymerase III must work along the other template strand in the direction away from the replication fork. This segment of the lagging strand are called Okazaki fragment. The ligase will act as a glue for connected the DNA strand that are missing. So, at the end of the process, there is two DNA. There will have two double helix DNA molecule. Is called as semi-conservative because it contains two strands, which is original strand and new strand. Thank you.